Now, for our next workshop today, we have Sarah Najad, who's going to talk about interplanetary synthetic biology. Super excited for this one. So Sarah, please go ahead and take it away.
Hi, everyone. So I'm just going to go ahead and share my screen and test that. All right. Okay, so I'm going to start with a really brief bio about myself. And then since we have only about 20-something people, if possible, I would like whoever wants to participate to just introduce themselves and talk about their discipline and also their interest in synthetic biology and space exploration, as well as what they hope to get out of this workshop specifically. So my name is Sarah Najad. I'm a recent graduate from NYU and a research affiliate. My background is in human-computer interaction and more recently biodesign. And my goal and my interest in my research is to really explore the convergence of biology, experience design, and space exploration. So I worked on different projects. It started with food, how to design the future of food, how to make the process more circular, more sustainable. And that led me to discover the potential of using microorganisms as a source. And of course, then down the road of synthetic biology as a tool to create different microorganisms to serve different purposes. So specifically, my thesis project was about the future of food in space exploration. The interesting findings that I had during my research was the challenges that come with space exploration, specifically long duration space exploration. And that is really high radiation, which leads to DNA damage, altered gravity, low temperatures, limited storage, and negative effects on health. And the needs that we have in the future, whether we're astronauts, scientists, engineers, or further in the future for space tourists, we're going to need transportation, habitats, life support systems that would produce food, oxygen, waste recycling, clothing, medicine, and other items. We also need a source of power, heat, light, and radiation protection. So the conclusion was that long duration space exploration depends on designing circular, regenerative, and self-sustaining systems. And biology could be this amazing technology that we can use to design such systems and grow different tools, food, smart fabrics, biofuel, personalized medicine, and even organs on demand. So why is life such an amazing technology? Well, first, it's programmable, so we can change it as we wish. Second is that it's modular, so it's easy to use one part for a different purpose and then repurpose it. It's self-repairing, self-replicating. It evolves depending on the environment or as we wish to program it. It in itself is a biosensor. It functions as a biosensor, and as well as this amazing built-in solar power. And it could be dormant for years and then be reactivated. So all these features make synthetic biology and biology in general a very attractive tool and resource for long duration space exploration. So in this workshop, we're going to kind of think about this future. I wanted to kind of limit this to 200 years from now, as we're getting closer to the NASA and also SpaceX deadline of 2035, which is the first manned mission to Mars. And so we need to design the future of these habitats and colonies and the technologies that we're going to use right now. And we need different kind of perspectives and people from different disciplines to come together to collaborate and see what we can come up with. So I wanted to, whoever wants to participate, I really would love to meet you and hear from you and see what your background is and how we can help each other out and design this future. So does anyone like to start?
I'm happy to start. Hi, Sarah. My name's
Hey.
Nikhil. My name's Nikhil. Sarah and I have collaborated on a number of projects, so I'm excited to kind of hear what the rest of you are thinking and what kind of cool ideas are brought to the table here today with all the different backgrounds you have. I'm a graduate student at ITP at NYU in my second year, focusing on very similar things to what Sarah talked about.
Thanks, Nikhil. All right, so okay, so let me tell you this though. This workshop is going to be a discussion and a collaboration. So I would highly encourage everyone to, if you don't want to speak up, that's fine, but the rest of the process is going to be a collaboration. So I would love for you to participate. Okay, so let's look at where we are on this timeline right now.
Sarah, quick interjection. Someone in the chat, David, had asked a question about what your specific research focus is in astrobiology.
My research is not specifically in astrobiology itself. It's in the intersection of experience design, interaction design, as well as biology. So as I said, I'm a designer and an artist. So my focus is to look at different perspectives, the human needs, but also the needs of other organisms, and see how we can design different systems that can serve not only us, but other organisms that will take with us as we explore space. So does that answer your question? Okay. Oh, cool. All right, great. Yeah. If you feel comfortable, you can unmute yourself. I would prefer this to be a discussion. So feel free to, cut me off and speak up as you wish. It's all good. I think that would be easier for me to interact that way.
I didn't want to cut you off, so.
It's no. Feel free to do that. My goal with this workshop is for it to be a discussion. I don't want to be the only one speaking. That would be super boring. So yeah. Do you have a question?
No, I don't have a question.
Okay. What's your background?
As I said on the chat, I'm doing my PhD at BU right now, but my previous job before I came for grad school was I worked as a space systems engineer. So I did some electronics work and integration work for them.
That's amazing. Okay, that's great to have an engineer's perspective on this. All right, so if no one else wants to introduce themselves, okay, so we can just jump right into the timeline. Go ahead.
I can introduce myself if you want me to.
Amazing, yeah, please.
I'm Patricia. I actually my background is in like protein engineering, and I actually do some work in astrobiology with Martian space yeast that can survive cold conditions.
Oh, great. Okay, perfect. So this is going to be perfect for you, especially with your technical background and perspective. You can really contribute a lot to this workshop. I really appreciate you being here. All right. Okay, so let's start. All right, so let's start from where we are right now. Year is 2020. What we have are these new trends and advances in technology, and specifically in synthetic biology. So we have these amazing tools like CRISPR, and we also have this increasing power in computation. We have the cost of DNA sequencing and synthesis going down, and then all these are going to make it more easy for people other than scientists and engineers to use this tool and create biology. So this is one of my favorite TED Talks from Durandi. And he really encourages us to think about what we can do with these tools and really use our imagination and not really focus on the more common uses of synthetic biology as we are doing right now. The other talk that I recommend you guys to watch is the second talk. It's all the uses and potential uses of synthetic biology for space exploration. And in this talk, they mention how we can reprogram not only microorganisms, but also plants and down the line ourselves to make ourselves more adaptable to the environment that we're in. So,
could you
yes.
We could see the titles and stuff.
I'm sorry, you were cut off.
Could you zoom in so we could see the
the
titles of the talks and stuff?
Yes, my bad. Do you also, do you guys have the link to this board? Did you get it? I dropped it in the chat. No? Let me do that again, just in case. All right. So now you guys should have access to it and be able to edit as you wish. Okay, cool. All right, so where were we? Yeah. All right, so right now we have the technology to really explore using microorganisms and re-engineer these amazing creatures to serve different purposes. So we can reprogram these microbes or yeast to create biobricks for us, for example, or create medicine, or be a source of food. They can create biomaterial. And we are also exploring how we can change the DNA of different plants and make them more resistant to harsh environments and hostile environments of the space. And so this can really grow, and we can see how this can evolve during this timeline. So down the line, like right here, we can re-engineer these microorganisms to help us terraform Mars, for example, and make the soil, or actually create soil for us that we can farm. And then eventually change the atmosphere of Mars and make it habitable for humans. Something even more wild would be to create this biological spaceship, for example, that would use asteroids for raw material and evolve itself depending on the environments that it's in. So these are really amazing prospects. I didn't really want to crowd the space too much, because what I really want and what the goal of this workshop is for you guys to really imagine and let your imagination go wild. And nothing here is right or wrong. And just because we don't have the technology or we haven't figured out the specifics of the science as of yet, doesn't mean that it's not achievable. So if it's in the realm of reality, even if it's far-fetched, don't hesitate to just put it down in the next step that we're going to get into. So for the next step, which is our first collaborative exercise, I really want all of you guys to look at all these categories. So we have like food, health, medicine, we have habitats, architecture systems, fabrication methods, we have fashion, clothing, production, tech, that includes the science as well. And we have this category for other. So just imagine the next 200 years of space exploration. So this could be anything from long duration missions. So we are on a spacecraft, or it could be on a different planet, a different colony. So just have that in mind. And then imagine what you want to see in terms of food, health, all these categories. What do you want this future to look like? What kind of foods do you want to have? what kind of habitats do you want to live in, what kind of new material do you see, and think about how synthetic biology can help us create these new items that your imagination comes up with. So let's just, you can either copy and paste the sticky notes, or you can just add sticky notes under each category. And do you guys have any questions?
Is the idea that we're kind of creating like mood boards down here?
Yeah, so this is like just rapid brainstorming right now. No idea is too wild or unrealistic. Just put whatever comes to your mind down here. You can also add pictures. I don't know if you guys are familiar with Miro, but I can just do like a really quick tutorial just to show you the main features. So if you look at the screen on the left-hand side, here you can add text, you can add a sticky note. You could also, if you go right here, so this is a great tool. You can use the Google image search if you want to drop in different images that you like. So for whatever item that you're thinking about, whatever image that kind of looks like or resembles what you have in mind, that would be very helpful as well. Do you guys have any questions? Okay, cool. All right, awesome. Okay, so it's really good to see all these cursors on the board. Okay, so we have 31 participants and only 12 people contributing. It would be really great to get your perspective, and especially like if you all come from different backgrounds. It doesn't matter if you're a biologist, an engineer, an artist, designer, whatever your background is, it's really important to have different perspectives, because depending on where we come from, what we've studied, what our research is, we're going to have our biases. And that's one of the challenges of designing for space exploration. The people who are designing for the space are a very specific group of people, and it's not diverse enough. So another point that was my objective with this workshop was to really create this diverse group of people and get this crazy like range of perspectives and points of views. So it would be great to see more people contributing. Nikhil, what did you add?
I bet you could guess. I added 2 notes, and I bet you can guess which one.
I bet I can. All right, let me zoom in, let me read. Real vegan cheese. Who added that? I'm all for that. Who added that? What do you mean by real vegan cheese?
Real Vegan Cheese is a community project with BioCurious and Counterculture Labs, where they're producing recombinant caseins from yeast for cheese making using actual milk proteins.
Amazing. So it has all the attributes of real cheese. So it stretches, it has the flavor, and it's all because of the certain protein that they're encoding, right?
Yes, that's at least.
Okay, cool. Would it be possible for you to also drop in the link so it's not confusing? Because honestly, I thought you just mean like real vegan cheese. I didn't know it was the name of an organization.
Sure, I wasn't the one that put it in the board, but I can provide a link.
Awesome, thank you. Meat in petri dish. Yes, lab-grown meat. Yeah. Yeah, so okay, so this is really interesting. When I was doing my thesis project, one of the most important aspects of my research was the experience of astronauts. So there's a lot of stressors on the astronauts. There's this psychological stress, and it's amazing that food can be not only a source of nourishment, but a source of comfort for the astronauts. So, and the range of foods that they currently have access to, it's not limited, but it's not the same as having the dining experience as we have on Earth. So they're limited to freeze-dried food or yeah, pretty much dry food that you need to rehydrate to consume, and they don't have access to fresh vegetables and fruits. So meat, I think they have access to that, but I would love to know if their experience of meat and the taste of meat that they have in space is the same as fresh meat. Oh, I also know of this startup, I forget the name, but they actually, so they produce lab-grown fish and they 3D print it, and they've done it in space at the ISS. So that's pretty cool. I can look that up and drop that in as well. Biofactories, absolutely, yes. Microbes for, yeah. So it's incredible how amazing microbes are. Yeah, so you can produce different foods, different material, and what's amazing about them is that the raw material that goes in for them to consume can be mainstreamed, and also they can be programmed to perform different functions, and they grow super fast. So they're very agile in a way, and very sufficient. So cool. Yes. All right, so that's another aspect of my thesis project. Use of microorganisms like cyanobacteria to transform CO2 in the atmosphere to oxygen. Absolutely, yeah. So when you're designing for space, it's really important to think about the system. And if you have microorganisms like cyanobacteria, so they could be a source of biofuel, they could be a source of food, but they're amazing because they actually consume carbon dioxide and produce oxygen. So if you integrate cyanobacteria into your life support system, it can have this multi-purpose function. Cool. Yes, Maggie, yeah, Interplanetary Cookbook, yeah. Yeah, Maggie is amazing. I follow her research and we exchange knowledge often, so she's amazing. I wish she could attend. Greenhouses for growing crops. Yes. I would like to, whoever added greenhouses for growing crops, it would be interesting to expand on that. So what would be the difference if we're creating these greenhouses? What's the difference between a greenhouse on Earth versus one, you know, on a spacecraft, or one on a colony on a different planet? Ice cream, yes. All right. Yeah, this is pretty cool because I think we're actually ahead of schedule. So we have plenty of time to add more and then go to the next phase. Yes, microbiome therapy, healthcare. Yes, yes. All right, so speaking of microbiomes, that's really interesting because there have been studies done about the microbiome of not only the spacecraft, I mean, at this point, it's just the ISS, but also the microbiome of the astronauts as well. And what's interesting is that the microbiome itself becomes less diverse, and the microbiome of the collective microbiome of different astronauts and the ISS becomes less diverse as well, and it starts to look very homogeneous and alike across the board. So finding ways to microbiome would be interesting and necessary, especially for the astronauts' health. Need to proactively increase microbiome. Yes, yes, yes. These are awesome. Yes, mental health. Yeah, so yeah, as I mentioned, psychological stress is a challenge, an issue in space travel, and especially in the future when we go on longer missions, that's going to become even more of a factor that we need to design for. So I would actually love to, if you have any suggestions for solutions, how can we address that? How can synthetic biology help us address that? That would be interesting to see. Yes, symbiotic relationships to prolong lifespan. So when you say symbiotic relationships, I'm curious to know, I'm not sure who added that, but I'm curious to know, do you mean between humans and microbes specifically, or other organisms? And if so, what kind of organisms do you have in mind? Do you mean plants?
I think that I meant within everything. I mean, I was thinking about just plants and microorganisms, but also humans and microorganisms, because I imagine that space traveling will take a lot of time, and we humans want to prolong their lifespan, so it's logical to want to address that issue.
Yeah, absolutely. So yeah, you hit on a really good point. So yeah, developing these symbiotic relationships with non-human entities, biological organisms, not only has a positive psychological effect on the astronauts or even space tourists in the future, it also creates these sense of stewardship for the organisms that we cultivate in space. And the truth is that we're going to immensely rely on these organisms. And if something goes wrong, that's it. So we don't really have access to any shipments from Earth to come to the rescue. So we need to be even more vigilant when it comes to stewardship and really thinking about these organisms and our relationships with them and the environments that we create for them to thrive in. And not just look at them as labor, as organisms that are there just to serve us, but as friends. Yeah, so having that symbiotic relationship is really important. Wow, I got a sensor suit. That's cool. You can also put that under fashion. 3D printer for implant scaffold. Yes, yes. The materials in yeah, yeah. Yeah, all right, cool. Yeah. Yes. So status, yeah. Yeah, that's interesting. If you want to expand on that, I would be interested to know more. Like, what do you have in mind? How do you see synthetic biology playing a role in that aspect? Yeah. Yes. Yeah. Okay. So this idea of growing architecture is really amazing and really appealing to me. So not only using just biomaterial, but engineering an organism to grow into your habitat, to become the habitat. So you don't even need to assemble it. That's pretty amazing. Air plants. Air plants. Glad at air plants, because I would like to know more. Yes. Yeah. So yeah, using bacteria or like microorganisms, so bacteria, yeast, fungi. Yeah, especially bacteria and yeast, that's really appealing, again, because they self-replicate so quickly. They don't consume a lot of raw material, and also they're easy, not easy, but easier to store. So storage is a factor, and that could be beneficial. Yeah, also like, so I have this picture here of this tardigrade. So this is crazy because tardigrades can go into a dormant form for years. And imagine if you use that feature for storage. Yeah, that makes using bacteria, like microorganisms in general, as your base material or base factories, really appealing.
Hi, Sarah.
Hi.
Excuse me. Thank you for sharing this project with all of you. It's clear to enjoy it, this project, and makes so happy for you. What do you think that should be the first steps to doing all this possible? Growing plants, or microorganisms, or another thing? What do you think?
Yeah, so realistically speaking, microorganisms are the first step. So it will be easier to reprogram them, test them, and see if it works or not. And then the next step would be plants, more variety of plants. And then down the line, we move to more complex organisms. So yeah, I mean, that seems to be the logical steps. But okay, so let's see, it's 5:05. All right, so if you guys are about done with this step, I'm going to talk about the next phase. Do you guys need more time?
Yes, I am more time to talk about all of this. I think that the answer was so cool.
Okay, cool. All right, so let's do, okay. Hopefully we're gonna have time because yeah, there's one more step to this exercise, and then there's the next exercise that I'm really excited about that one as well. So, and I wanna have enough time. It's 5:06, so we have until 5:45, I believe. All right, so let's do this let's wrap this step up in the next 2 minutes. So we have enough time to do the rest of the activities. But this is super cool. So many amazing stuff. Yes. Yes. Space plants, super cool. So when it comes to imagining these novel organisms that we haven't created yet. It's fun to picture them, right? And so what I'm going to do, I know we're not going to have enough time to do that yet, but what I'm going to do is to have the board available for you guys to edit until Monday. So when it comes to those things that you may have this picture of it in your mind, and maybe you want to do like a quick sketch, or maybe you want to find more pictures that would kind of resemble what you have in mind. You can add that to the board later on too. So that'd be super cool, because yeah, space plants, I want to see that too. And I want to see what you guys see in your head when you think about space plants. So like, how do they grow? Like, to which direction? Where do the roots grow? To which direction? There's been studies done, but that's very limited too. We don't know how that's going to change in different levels of gravity. So in microgravity, apparently that doesn't have much of an effect on the way that plants grow. They grow towards the direction of the light. So that's the basics of it. Yeah, but it would be interesting to like think like, how about on Mars? How about when we achieve interstellar travel in a different galaxy? What would that look like? Yes, closed symbiotic systems or microbes. Yeah, so, okay, this is super cool because it kind of confirms what my thought process was when I was doing my thesis project. And I've actually linked it to, let me show you. So this picture right here, that is linked to my thesis project. And what it is basically this concept for an installation that depicts the future life support system or habitat in space. And it actually incorporates this symbiotic system that uses, it integrates different microorganisms like cyanobacteria as a source of food, as a source of fuel, biomaterial, and different kind of microbes that then recycle the waste, the organic waste. So the cyanobacteria also produces, also produces oxygen, which is needed as a feature of the life support system. So if you're interested, you can check that out later too. But this is awesome. Okay. Oh, okay, so let's wrap it up. Cool. And you can always add to this until Monday, that is. All right, so for the next part of the exercise, I want you to either look at the items that you added, or if you see something else that is interesting to you, and when you, let me show you how it works. So when you click on a sticky note, do you see these dots, the blue dots? So if you click on that, here we go. If you click on that, you can actually draw a line. Let me change that to white and increase that. Okay, so you can connect that to a point on the timeline. So in this step, I'm interested to see when you predict this specific item to become reality. Does that make sense? If it makes sense, go ahead, add it. Oh, something happened here. Because I believe the timeline was moved a little bit. I'm just going to change that. Okay, cool. All right, so again, the timeline is from 2020 to 200 years from now. So you can connect your sticky notes to any point on the timeline. And while you do that, I'm just gonna read more sticky notes and see what ideas you guys have. Yeah, so microorganisms and microgravity. Yeah, so that's something that's being researched on right now. So the effects of radiation and microgravity on different microorganisms. And again, this is, okay, so when it comes to, as I mentioned before, so one of the challenges is different levels of gravity. So it's a specific term, I forget, wait. Altered gravity, it's called. So yeah, it's not just microgravity. It could be different levels of gravity depending on where in the universe we are. And it's, yeah, we don't know. We don't know how different levels of gravity is going to affect different organisms. And it's important to keep that in mind. So when we're thinking about synthetic biology, the kind of tools that we're going to use, the kind of interfaces, the interactions, we need to take those factors like microgravity, altered gravity, into account as one of the materials that will affect the tools that we're designing and will use. So we're going to get more into that in the next step of the exercise.
So I wanted to ask something. So in a lot of these cases, we think about biology as conventionally like what we find on Earth, right? Where there's an abundance of water and air and whatnot. But in space, that's actually a limited resource.
Absolutely,
so do we still consider it the same way? Like, for example, if we grew a plant on Mars, or if there was a Mars plant with the current atmospheric conditions, it wouldn't be the plant we have on Earth. It would be something that would actually survive in that ecosystem. So I started thinking when you said, like, if you try growing plants in space, they just move towards the light. They don't really care. Gravity doesn't affect it as much. So I'm trying to think.
Can I just add something? I said the research so far. So that's not absolute. That's not a fact. Yeah.
I mean, putting that as a hypothetical, right? Like, if that is the case, then what would biology optimize for? Like, that's what I'm trying to think of.
Yeah. So yeah, it's interesting that you mentioned that. So one of the research that NASA, for example, is doing is to develop plants that are more resilient to hostile environments. So they're more drought resistant, for example, or radiation, or they can survive in lower temperatures. So we can engineer the plants to respond to the kind of environment that we're in. So this could be specific to the planet. For if we're like on the moon, or if we're on Mars, we're going to have different sets of plants, right? Each tailored to their environment. But another approach could be to really kind of perhaps accelerate evolution. So instead of us just looking at the environment and specifically engineering that, what if we could engineer organisms to respond faster to changing environments and adapt faster? So in a shorter span of time, what would that look like? Is that a possibility?
Yeah, that might actually solve some of these questions, right? Like, I honestly don't know what plants are optimized for in space because it's but if, like, like when you were talking about what it is. I'm also, I guess, one mistake I was thinking was I was thinking of plants alone in like a sandbox, but there are humans and other organisms over there. So there's a more interaction. So they're probably optimized for whatever suits best with those other organisms.
Absolutely. Yeah. So yeah, we're pretty much going to design them specifically for the environments and also kind of guide their evolution in a way as well.
Yeah, I mean,
Nice.
I think it'll make a great dystopian sci-fi setting where, we let them engineer themselves and they decide all humans are useless, so let's kill them off.
Yeah, it's so interesting because especially like when it comes to synthetic biology, because it opens up so many doors and it kind of makes the science fiction possible in a way. We always think about this dystopian futures, and I know because it's exciting, right? I mean, I've done that too in my project. Like in one of the projects that we did, Nikhil and I and other collaborators worked on, we created like short sci-fi about this colony on Mars. Nikhil, do you want to tell the story? Keep it short though, because
it's such a bizarre story, but we basically imagined a future where a sort of like pansentient microbial mass figures out how to communicate with like basically with human cells and like causes tissues to disassociate and break into constituent like individual cells and reform into a pansentient mass. Sorry, I don't even know if I can explain this thing in a short way.
That sounds amazing. You basically took cancer and and microbiomes.
Yeah, it was very dystopian from a human perspective, but from that pansentient perspective, it wasn't necessarily. So yeah. But yeah, I mean, I try to be not a utopian futurist, but I try to imagine what I actually want to see happen, like the future that is desirable for me, and then work towards creating that. As exciting as dystopian futures are, I think like if we pay attention to them more and focus on them more, then we don't really dedicate the mental and creative capacity to the futures that we actually want to see realized. So that's my philosophy personally. But yeah, dystopian futures are cool too. All right, okay, so it's 5:21. I think it's, yeah, we can move to, if it's okay with you guys, because the next exercise is pretty cool too. You're gonna like it, because I really like it. Okay, so are we ready to move to the next phase? Yes, no, yes, no. Yeah, thumbs up. Okay, cool. And again, like you can edit later too. So this is going to be available to edit. All right, so for the next part of the workshop, the second exercise. So this is super cool. I mean, I personally really like it. This has moved. I should have locked these. All right, so for the next phase, you're really going to like imagine a scenario and go with it. So for this, I want you to pick a character for yourself or a persona. And this could be anything. So you can picture yourself as a scientist, as a designer, as a future space tourist, as a CEO of a company that is basing a new colony on Mars, or it could be like down the line as this transhuman being. So yeah, so your options are open. I want you to have this persona in mind. Think about the when. So on this timeline of the next 200 years, or if it's beyond that, that's okay too. Think about the when, think about the where. So where are you? Like, are you on Mars? Are you on the moon? Are you in a different galaxy? Or are you on a mission? Like, are you actually on a spaceship traveling between different planets? So that's up to you too. Think about the where, and then think about the what. So you're this person, like, let's say, okay, so I'm this criminal, and I'm on this colony on Mars, and the year is 2100. And now think about the what. So the what could be anything. So think back to the previous exercise, to the items that we just imagined. So it could be food, fashion, technology, a habitat, anything, any kind of tool, anything that you can imagine that could be created with synthetic biology. So to create this, what kind of tools do you need to use? What does this interface that you use to create this look like? How do you interact with the technology at that point in time? What kind of raw materials do you need to create that and kind of create this new transformed object or even organism? So it could be anything. What kind of raw materials do you need, and how long does it take you to make it? So the speed of actually synthesizing and then fabrication, that's a factor too. And then how do you use this item? So yeah, so think about all of those. And yeah, and also, okay, so you can add images that are inspiring to you and related here in the box on top. And yeah, so that's it. This is the first part of the exercise. So let's do that for, it's 5:25. Let's do it for 7 minutes-ish. Do you guys have any questions about this exercise? Is it clear? No? No questions or not clear? I really need to look at the chat. You guys, I don't look at the chat because I'm not that good at multitasking. All right, let's see. I promise I'm going to read them, all the comments soon. Okay, no questions. Oh, I'm sorry. Alexandra, if you're still here, I'm gonna, wait, someone already dropped it. Thank you, Nikhil. Thank you so much. Like,
good, I got it. I'm filling out my character.
oh, thank you. I'm so sorry. I know, like, it's my responsibility to be on top of the chat as well, but it's just not easy. So thankfully, Nikhil came to the rescue. All right.
Thank you, Nikhil.
Anytime. We're all in this together.
Ooh, yes. Okay, so I just thought, like, this is like a random comment that I picked up on. So the legal, okay, we're gonna get into that in the next phase. Yes, there is a treaty. Is it reinforceable? Who knows? I mean, I don't know which organization it was, but they actually, so we're not supposed to contaminate other planets. And there was this organization that kind of spilled tardigrades on the moon, I think. I'm not sure, like, I need to fact check, but this is what I remember. But yeah, I mean, based on the treaty, we can't do that, but is it reinforceable? The laws aren't actually laws, they're agreements. So which calls for really like thinking about all the consequences of the kind of technologies that we're designing, right? So, and we're gonna address that in the next phase. TED Talk. Esther. All right, which TED Talk? If you, I mean, they're all embedded. All the TED Talks that I included are embedded. So if you click on them, you should be able to view. Like major. Okay, thanks, Lucas. All right, so the organization, again, I'm not sure which organization it was. So what kind of punishment did they face, do you know?
Hey, yeah, so I'm talking about specifically the UN has the Moon Treaty and the Celestial Body Treaty, both of which, the first of which bans nations from claiming celestial bodies like the moon for their own, which is weird. The second one is the more pertinent one here, which is it prevents you from polluting celestial bodies, which is actually why, and it's not really, I don't think it has any punishments built into it. It's just basically a bunch of people from the UN going, hey, good thing to not do right now.
Exactly.
It was signed in like the 70s or 80s though.
Yeah, see, that's the thing. So yeah, we're not supposed to pollute it, like especially with any live organisms, any like earth biology. And yeah, it's an agreement. And if someone either intentionally or accidentally break this, which they've done already on the moon, there isn't any kind of specific punishment or consequences as of right now that is enforceable that I know of. So yeah, especially like I think with synthetic biology, it's really important to think about all of that. So if we're looking for example, like with astrobiology, if we're looking for extraterrestrial life or like microbial life on Mars, for example, if we contaminate it with our own microorganisms or with synthetic organisms, how are we going to know if it's Martian or if it's not? If we actually discovered life, or if it was just, you know, a consequence of our own action. So we need to think about those. Like we need to have markers on those new organisms, for example, so we can identify. So that's like one of the solutions that I know of so far to address that problem.
Well, I guess that also goes into how exactly you're looking for life. For instance, we have found traces of life on Europa, but that trace is just methane. So it could be that you, even if you make organic life and put it on a different planet and then mark it, the byproducts are how you look for life. So maybe you'll be finding the byproducts which can't be marked.
Yeah, that's a great point. So how do we actually identify life, right? Like what do we look for as a sign or a biomarker to know for sure, for a fact, that there is life on a different planet, for example. So, and that's what's interesting is that, okay, so how can we, to make it like more specific to this workshop, how can we think about synthetic biology and use that to create more accurate biosensors that are not maybe prone to the same inaccuracy, right? Is that a possibility?
I don't quite think so, because a lot of the sensors we use currently aren't really biosensors. Like if you're looking through a mass spec, you don't really find a sign of life. You can't tag a carbon for being your carbon. It's just bulk carbon.
Yeah, but can we? I mean, as of right now, we don't, but can we? Can we actually develop biological biosensors?
That's a question for someone who's paid more than I am.
I guess the question is, how do you define biology for that? Because as, I don't know who was speaking earlier, was saying, like, if you use any physics-based methods, you just identify what molecules or what atoms are there, and that's all.
Yeah.
So if you want some other more complex structures, so that's why I'm asking, like, what is your definition of biology? Like, should it have DNA, or should it have RNA, or can it be something even more primitive than that? And that kind of formulates whether or not. And in the cases of like in signs of life, I believe they find traces of amino acids and stuff, and which is why they can do. I need to check on that, but
yeah, that's a great question too. Like, how do we actually define biological life? That's from our own very limited perspective as, you know, human beings, so, or earthlings in general. I just saw Nikhil's comments on asking people to introduce themselves and talk about their backgrounds. Yeah, it was super cool. I mean, if anything, I can add a list to our Miro board if you're interested for like future collaborations, research in this specific space and integration of synthetic biology and space exploration. You can just, you know, type your like name, email address, like the best way to contact you, and also your background, your research focus. So that would be an interesting resource for all of us to have access to. What was that? Sorry, did I cut you off?
Yeah, Chris invited me to his house.
I think someone's mic is juggling. Amazing. I just saw Christo's comment. That's a very interesting area of research. So his focus is specifically on using extremophiles and synthetic biology as a way to terraforming Mars. So pretty cool. Yes. Yeah, so yeah, Lucas, your comment about the taste in food, that is correct. So your experience of food is not the same. This is like Maggie's, I'm not sure if you're familiar with Maggie's research. She's at the MIT Media Lab in the Space Exploration Initiative. And her research is all about creating better culinary experiences for the astronauts and also future space tourists. So this can be anything from enhancing the food, I would say scent, to kind of enhance the flavor, but it could also be designing a different kind of tool to consume food in a way that you're more exposed to those molecules. So You can smell the food better. Therefore, you can taste it better. Therefore, your experience is enhanced. So I suggest that you look into Maggie's research. Let me search for the link. There we go. And she's pretty amazing. All right, ooh, we're running out of time, so I'm not sure if I answered all the comments and questions. Okay, how are we doing with this part of the exercise? Are we almost done? Because there's one more step that I think is very crucial, and we have less than, wait, what time is it right now? Ooh, we have less than 6 minutes. Okay, are we ready for the next phase? Yeah. Again, you can edit this later. As long as you have the main idea, you can elaborate on that later and you can add more details. So, okay, so for the next and final phase, I want you to, if you can see my screen. Okay, so if you see the table that I put on the bottom, this is for a SWOT analysis. So we're going to think about this new object or entity that we created via synthetic biology, and then think about the strengths of it, the weaknesses of it, the opportunities, and also threats. So this is like what's important when it comes to our role as designers and scientists and engineers, because we can get really excited about an idea and the potential of it and lose sight of the consequences, you know, some that we may think about, but some that we're absolutely oblivious to. So I'm going to, okay, put these here. Oh, what happened? Oh, okay. All right. Cool. No. All right, so now you can just add sticky notes, if possible, with the same colors. So green for strengths, orange for weaknesses, blue for opportunities, and red for threats. And add that about the item or the organism or the technology or the tool that you just created. it's really cool to kind of read everyone's short sci-fi stories here. One thing that I'm noticing is that I know like we're short on time, but we think about these ideas, they're very almost like big picture ideas and not very specific. It would be interesting to, once you have the main idea, to then really think about the details of it. So yeah, like I'm really curious to know how do you go about, like how does your character go about, it's like, oh, I need this, so what am I using to create this right now? Like, is my interface a hologram that I design in, and then the output is biology, and then it's fabricated by a 3D printer? Like how does it work? And it depends on your timeline too, right? Like on the year that you pick. So, and like, how do you interact with it? Let me fix this.
What's the blue post-it again?
Opportunities. Okay. So I just I just like the background so it doesn't get in your way when you move things around. All right. Okay, so we're running out of time. It's almost 5:45. All right, so again, feel free to edit tonight or anytime until Monday. Okay. I'm going to add a form for you to write down your information, especially if you're interested in collaboration with other people with different disciplines who are also interested in this intersection of fields. And also, if you have any questions, here's my email. If you want to look at my projects, you can go to my website. This is my Instagram handle. It's mostly my art, but you can find me there. And yeah, with that, I want to thank all of you again for participating. It's really amazing to see everyone and like really learn about your ideas and how you imagine this future to look like. And I'm also looking forward to getting your information and possibly organizing more workshops and events in the future, maybe more possibilities for us to collaborate. So do you guys have any questions before we wrap it up? Also, I really hope you guys had fun with this. That was my main goal.
Everyone's so quiet. I guess they're still having so much fun in the mirror board, Sarah. They're just so caught up in it.
I mean, no, I get it, because I participated in workshops like this before, and it is not easy to multitask. I'm telling you, like once you get excited about an idea, you just want to write it down and you want to like make it really like well formed. So I don't blame them. It's all good.
All right,
and again, so they're going to have access to this until Monday.
Yeah. So I guess like if people could come back, if everybody's okay taking a quick family photo so we can document this awesome experience. But Sarah, thank you so much for sharing this and through guiding us through this exercise. This has been enlightening. I feel like we take being a part of something like big here that is going to come up in the future over and over and over again. So I look forward to seeing how this develops. I think an idea for—
Thank you so much for organizing, and thank you everyone for participating.